Good afternoon. I'm David Ackerley, Dean of the Rausser College of Natural Resources at UC Berkeley. The Department of Plant and Microbial Biology is one of the 5 departments in the college. PMB is pleased to announce a series of events and programming in honor of the department's 30th anniversary. Like all of you, we had hoped these events would be in person, and hopefully we will come together in the future. And for now, we'll come to you virtually and hope we can reach a really broad audience that way. To commemorate its 3 decades of research, teaching, and service, PMB is hosting a series of online talks through 2020 that cover topics which reflect the department's deep contributions to fields that include the basic biology of plants and microbes, applications of CRISPR-Cas9 to agriculture, innovation and entrepreneurship, and close ties between industry and academia. Please visit the department's website at pmb.berkeley.edu to watch for announcements of future events. You may have heard the inaugural talk on June 12th titled Of Virulent Viruses and Reservoir Hosts with PMB professor Britt Glossinger, as it was featured on the Berkeley Conversations COVID-19 series. The talk was recorded and can still be accessed at the Berkeley Conversations website. Today's talk is on the future of food. I'm pleased to moderate today's panel discussion with 2 people who are very well known to the PMB and Rausser College community. We'll be taking live questions via Facebook Live or YouTube Live, whichever format you may be listening in. And we'll come to those a little later in the session. We're also recording the talk and it will go live shortly after we wrap up today. Brian Staskawicz received his PhD in plant pathology here at UC Berkeley. He is currently a professor in PMB and the scientific director of agricultural genomics at the Innovative Genomics Institute. Brian has made many seminal contributions to the understanding of infection strategies, of plant pathogens, and defense mechanisms of plants. These include the cloning of the first pathogen effector gene and the cloning and characterization of one of the first plant NLR immune receptors. Brian and his colleagues also played a major role in establishing Arabidopsis as a model system to study the molecular basis of microbial recognition by plants and genetically dissect defense signaling pathways. More recently, he's leading an effort at the IGI in genome editing of agricultural crops for biotic and abiotic stress resistance and improved plant performance. In 2013, Brian was awarded an honorary doctorate from Wageningen University in the Netherlands. He is a member of the US National Academy of Sciences and a foreign member of the Royal Society. In addition, he has been elected a fellow of the American Phytopathological Society and a fellow of the American Academy of Microbiology. In 2019, Brian was awarded the International Society of Molecular Plant-Microbe Interactions Senior Investigator Award for his many contributions to the field of plant immunity. Pam Ronald received her PhD from Berkeley's PMB department in molecular and physiological plant biology. She is recognized for research in infectious disease biology and environmental stress tolerance. Pam was a postdoctoral fellow in plant breeding at Cornell University and joined the faculty of UC Davis in 1992. In 2019, she was awarded an honorary doctorate from the Swedish Agricultural University. She's a faculty science at Berkeley Lab's Environmental Genomics and Systems Biology Division and a key scientist at the Joint Bioenergy Institute. She's a faculty affiliate of the Center on Food Security and the Environment at Stanford University. A National Geographic innovator, Pam was named one of the world's most influential scientific minds by Thomson Reuters and one of the world's 100 most influential people in biotechnology by Scientific American. She is a member of the National Academy of Sciences. She is co-author with her husband, organic farmer Raoul Adamczak, of Tomorrow's Table, Organic Farming, Genetics, and the Future of Food. Bill Gates calls the book a fantastic piece of work and important for anyone that wants to learn about the science of seeds and challenges faced by farmers. Her 2015 TED Talk has been viewed by 1.7 million people and translated into 26 languages. She founded the UC Davis Institute for Food and Agriculture Literacy to provide the next generation of scientists with the training they need to become effective communicators. In 2019, Pam was awarded the American Society of Plant Biologists Leadership in Science Public Service Award. Pam and Brian are here to discuss the role of new agricultural breeding technologies in the quest to feed the world's growing population while enhancing the sustainability of our agricultural practices around the world. In a world approaching 8 billion people, with most of the available arable land already in use for agriculture, and the threat of a rapidly changing climate, this challenge has never been more pressing and immediate. Brian, can you set the stage for us by describing the scope of the challenge in front of us in relation to the work you do and the topic of today's discussion?
Great, and welcome to everybody this afternoon. It's a pleasure to join in this panel. As David said, the world's population currently is about 7.9 to 8 billion people, and it's predicted by 2050 to reach 10 billion people. And unfortunately, we have to basically grow food more than 70% increase in production during this time period to feed the world. Unfortunately, also, that this is exacerbated by climate change. So being able to grow this food in an environmentally sustainable fashion is going to be extremely important. As I mentioned, climate change really will have a major impact on disease pressures in plants. We already know that pandemics such as wheat blast are occurring in Bangladesh and India because of various jumps in hosts. In addition, many plant disease resistance genes are also temperature sensitive, which, as you can imagine, as the climate increases, the temperature increases, the effectiveness of these genes will be lost. So to meet these challenges, new technologies will be needed to increase food production. In addition, high input resource intensive farming systems, which have caused massive deforestation, water scarcities, soil depletions, and high levels of greenhouse gas emissions, cannot deliver sustainable food and agricultural production. Obviously, there are no simple answers to these problems, but we have to find a way to produce food in an environmentally sustainable fashion. That is, we need to decrease farmer inputs such as water, fertilizers, and pesticides. I am optimistic that we are developing the tools to accomplish these goals, and this will be the subject of the talk today. Thank you.
Thanks, Brian. Pam, turning to you, can you elaborate more on what it means when you think about making agriculture sustainable locally and globally and into the century ahead? And put that in the context of these very fundamental challenges that Brian has laid out.
Yeah, I think Brian pointed out some really important goals for all of us. Using land and water more efficiently is going to be critical. Most of the arable land has already been farmed, so the land that we have, we need to use much more efficiently. And Brian also mentioned reducing harmful inputs. There are other challenges such as maintaining soil fertility will continue to be really an important goal for farmers and maintaining farmland. We have to think about farmers themselves, how they can thrive economically, not only in the United States, but in other parts of the world. And finally, we really need to be sure that we can keep food affordable for even the poorest people in the world. And I also like to thank you for including me in this conversation. And I'm really pleased that to be here and appreciate the audience for joining in.
Thanks, Pam. Brian, I know when you teach here on campus, you paint a picture of the broad sweep of the history of agriculture for our students and putting our modern challenges in context. So if you go back to the beginnings of agriculture, farmers and plant breeders have worked together and in parallel over many centuries to bring us the crops that we have today. Can you share a little bit about that history of crop breeding and how it brings us to the technologies that are available to us today?
Absolutely. So as we have discussed in my classes a lot, all plants, many people don't know this, that have a geographic origin in the world. For example, corn or maize was originated in Mexico, rice and soybean in Asia, and wheat in the Middle East. So these are distinct areas. Agriculture is considered to be approximately, and depending on the studies you read, between 10 to 14,000 years old. And all our modern crops rose through the domestication of these wild relatives that originated in these parts of the world. As plants were domesticated, plant breeders deployed these ancient varieties as sources of traits that they introduced into modern day varieties by classical plant breeding, meaning taking pollen from parent A and putting it to parent B and getting progeny from that. So just classical plant breeding, making genetic crosses. In fact, plant breeding as a science was only developed in the early 1900s. And as we know it today, has become extremely sophisticated and deploys modern tools of DNA marker-assisted breeding, and also employs crop genomics. But unfortunately, plant breeding is very inefficient and takes many years to introduce these traits into our high-yielding agronomic varieties that we use today. Thus, new breeding tools that we will discuss today will be needed to breed varieties faster and more efficiently so we can adapt to the climate changes that are occurring throughout the world today.
And we will definitely circle back and talk more about the actual, the mechanisms of what we mean by those new technologies. But Pam, maybe over the course of your career, if you think over the course of your career, can you just give us some examples of what it means as a researcher to be on the ground, so to speak, or in the lab, applying these technologies, or the kinds of traits that you've worked on, the crops you've worked on, and what it's meant to be a scientist involved in all this work?
Well, it's been really fantastic to be involved in plant biology for so many years. And I had the privilege of working with scientists at the International Rice Research Institute really from very early in my career. So I was trained in Brian's lab, lucky me, and learned a lot about disease resistance. And when I began my first postdoc and first position, I worked with Gerd of Kusch and others who had discovered a wild species of rice that had a really important gene for resistance. And so many of the resistance genes that we use and that are in our crops have been derived from some of these ancient varieties or diverse species. And so that has been something I spent a lot of time working on, trying to understand the molecular basis of disease resistance in this wild species of rice from Mali. And then more recently, I had the honor to work with David McHale, who also had, he's at UC Davis, Cornell with me, but also had spent some time at the International Rice Research Institute. Brought to my attention challenges faced by farmers growing rice in many parts of Asia, primarily Eastern India and Bangladesh, who often lose their crops to flooding. So flooding is predicted to be increasingly common as the climate changes. And most rice varieties will die if they're completely submerged for more than 3 days. So Dave and I were able to isolate a gene that conferred resistance or tolerance to this flooding. And the International Research Institute, led by Dave and his colleagues, were able to introduce this gene into varieties grown by farmers in Bangladesh and India using marker-assisted breeding, which Brian mentioned, which is a genetic technique that's not regulated and has been really important for developing new varieties. So this variety does really fantastic. So even after 2 weeks of flooding, the plants can survive and thrive. So the plants that have this gene have about a 60% yield advantage. And this is really important for farmers in Eastern India and Bangladesh who are able to harvest grain even in the face of severe flooding. And last year, about 6 million farmers grew this rice, which is called sub-1 rice.
Pam, I want to follow up briefly on a comment you just made, because you talked about the traits that are important for farmers. And then, of course, for those of us who are on the other end eating the food, the question is, what are the traits that as consumers we're looking for? So as researchers and as plant scientists, what is that balance of the traits that farmers want and the traits that consumers want in the ones that have drawn attention in the research community?
So one thing to keep in mind is for a crop like rice, the farmer is the consumer. So most people that are growing rice in less developed countries are subsistence farmers. So that means that they grow really only enough rice to feed themselves and their families. So when a farmer has the ability to grow rice even under flooded conditions, that means that they're able to feed their families and sometimes even sell some of that rice to bring in some income that they can use to support their children's education, for example. And then if you're in a developed country and are possibly not concerned with how farmers are able to grow their foods, so there are many different types of traits that consumers in developed countries are interested in, and particularly people are interested in color and flavor and diversity and taste, of course. So, but those are also concerns of farmers in the less developed part of the world as well. Is that what you meant?
Yeah, well, absolutely, and of course, what consumers want is of concern to what farmers are growing, because it's an integrated system, and part of sustainability is financial sustainability. And if there's not what does the market want? And if there's not a market, then that's going to impact, of course, practices on the farm. So the food system might be one of the most complex parts of our modern society. That entire chain, right from the soil all the way through to the waste product at the other end. So lots of moving pieces. So you've been talking about rice, and Brian, I want to talk a little bit more about the diversity of crops. So in the northern hemisphere, or perhaps almost across the entire globe, a huge majority of the calories that we all eat come from just a few crops, and corn and soy and wheat and rice, I think, top that list. But of course, around the world, people eat a whole lot of different things, and of course, we all enjoy diversity in our diet. So in your work in the Innovative Genomics Institute, I'm curious what other crops you're working on and what the motivation is. Like, what are the crops that really need attention as we face the challenges of the 21st century?
Absolutely. So the IGI, which is the Innovative Genomics Institute, which was started a few years ago by Jennifer Doudna, basically, since it's a lot of philanthropy funds our research, we decided to work on crops that really impact a lot of the developing world. And I think it's fair to say that CRISPR technologies, or gene editing technologies, will probably have a greater effect on billions of people in the world, where in medicine, maybe hundreds of thousands. So I think it's quite evident that I think that using gene editing technologies in agriculture is going to be extremely important. So we set out a very robust plant transformation DNA delivery systems, and we established crops such as wheat and rice as being important. And we're able to actually regenerate plants, and not just the model plants, but the ones that farmers actually use in the field. But I think a couple of other examples that we're currently working on is that we work on cacao. People that like chocolate are interested in cacao. What you should realize is that 50% of the world's chocolate is produced in the Ivory Coast, or Cote d'Ivoire, and that it's subject to this really important virus disease called cacao swollen shoot virus. So we are working in collaboration with Mars Corporation, who gave us a nice gift to actually study how we could actually use gene editing to alleviate this particular pathogen from cacao. So very important, and it's a very challenging crop. We have very recently about ready to submit a manuscript where we've actually accomplished with Myung-Jae Cho, who's the director of plant transformation and genomics, the ability to actually manipulate cacao plants in tissue culture. So that's a great advancement. Another crop that we work on that companies don't work on is we work on cassava. And cassava is a plant that originated in South America, but it's grown a lot in Africa, so smallholder farmers. And one of the major problems with cassava is that a lot of the roots produce cyanide, and they're very toxic to the people that eat this, and they can cause various diseases in these smallholder farmers when they eat these various crops. So we have developed systems now where we have identified the genes that are involved in making cyanide in cassava, and preliminary results suggest that we have eliminated cyanide production from these plants. And so this is a great accomplishment. We hope to be able to field trial these in the near future. So cassava is a plant that many people, as I said, but it's not a cash crop. So you don't find companies working on these. So we feel that an institute such as the IGI, who's got these resources, can apply these to crops that are important for the developing world. You know, we work on tomatoes also, where there's some diseases that we're involved with, but major crops are wheat, rice, cacao, and cassava that we talked about.
Yeah, early in my career, I had the great opportunity to spend quite a bit of time living in Brazil and working in the Amazon, and ate a lot of cassava because that becomes a real staple. And just it's something that's on the table in various forms, you know, with every meal, and something that's not part of our diet at all in the Northern Hemisphere in that way. Brian, I want to stay with you for a moment, and because you've mentioned CRISPR, we've talked about CRISPR, you've mentioned gene editing. For our broad public audience out there, most people just know the term GMO, and it's just this one word, and it's a single term, and yet it encompasses a very wide variety of underlying technologies and biological discoveries. So I think this is really an important moment. Just can you share from, especially in your work with the IGI, how to make sense of that term GMO, and then what does this word gene editing mean? Like, because that is, of course, we're hearing this much more in popular discourse and in the media.
So typically, a GMO is a plant that is created by using plant transformation with a vector called Agrobacterium tumefaciens, which is a natural soil bacterium that transfers its DNA to plants. And scientists in the 1970s, the early 80s, found a way to disarm these particular plasmids and insert genes of interest and then put them into plants. Now, one of the criticisms people have had about GMOs is that when you insert DNA into plants, it goes into a random part of the genome. So we can't control that. Now, gene editing is an invention that actually it's a natural system that bacteria have used to fight bacteriophages or viruses that infect pathogens. I mean, these phages that come in, these bacteriophages, and the bacterium can actually eliminate them by recognizing the DNA and then cutting it into small bits. And so essentially what CRISPR allows you to do is to do more precise gene insertions into the place. So we can now direct and target genes into the plant in a very precise way that we couldn't do with GMOs. And that, in fact, in the United States, APHIS, which is the Animal Plant Health Inspection Agency, the new rules and regulations say that plants where we make simple insertions or deletions are not regulated as a GMO, and they don't pose any greater risk than would naturally be done. So
I want to just to continue with that moment. One of the analogies that you often hear is that the genome is like the book, a book, and what's happening in evolution is the letters are changing and the words are changing, and some of those mutations or it's like a typo have no meaning at all, and then some are quite important. So it sounds like in some cases what these technologies have done is like taking a whole section of a different book and moving it in, like just copying and pasting, versus the equivalent of a word processor of making a correction, a change here or a change there. Am I pushing the analogy too far? Does that help?
No, I think these analogies have, you can think of it as a word processor. So for people in the audience that aren't familiar with this, that some of the applications that are being done in biomedicine, so for instance, they've been very successful now. There are trials out there right now where sickle cell anemia has been identified by a particular mutation. And so scientists can go in now and basically change the gene very simply by using CRISPR technology to change that. So we can make precise changes. We can change that basically correct a bad allele for a good allele. And the same thing could be done in plants. We can do that. But also in plants, we're hopefully going to be able to, and Pam can talk about this later, insert blocks of genes into plants to actually, that Pam has done for with vitamin A. She can talk about that maybe too. Yeah. So I think it's those are good analogies for people. So it's really the precision that we're able to do with this. And at the end of the day, we can actually tell people these are the precise modifications that we've made, as opposed to GMO, you're kind of don't know where it goes into the plant.
And then I suppose the other important difference is gene sequencing is now so powerful that after making the change, you can verify, right? You can verify what actually changed and know that the background is more stable. I mean, I know Pam, in your book, you just gave the anecdote of something that cost millions of dollars just 15 years ago. The genome sequencing is now, just $1,000 or a few $1,000s. The pace of change in this area has been truly breathtaking. Now, Pam, when you talked about the rice, you mentioned bringing a gene from wild rice. So clearly, in some cases, we need to know where can we find these traits is an important question. And I think there's a lot of recognition that the wild relatives of our crops are incredibly important sources of material. So we really depend on the maintenance of those wild varieties as well as trying to develop more productive crops. So that's a specific question, but maybe if you can just think about that, but also just put in broader context, what's the role of biological diversity in developing our cropping systems? And I'm going to feed you a second part to this question. Your husband's an organic farmer. Some people might find that a bit unusual to have an organic farmer and a plant breeder. It's working side by side, and you've written a book together. Because the other part of the diversity question is the role of diversity on farms, and series of questions we've been addressing for decades about the potential vulnerability of monocultures, the value of diversity within a farming system. So I'm just a couple of different questions about the nature of biological diversity and the role it plays in agriculture.
Yeah, well, as you pointed out, there's biological diversity on farms in the sense that you don't want to just plant one variety on massive acreage. And it's nice to sometimes put in different species, intercropping. And so there's a lot of examples like that. And then for geneticists, often biological diversity means bringing in different genetic materials. So to make your line more diverse, more resistant to disease. And we scientists for many years have been bringing in genes from wild species. So there is transfer between species that's been going on for many years in breeding. So that's very, very, very important, because if you can bring in a diverse set of genes, then you're going to be less vulnerable to epidemics. There's also biological diversity that you can bring in from outside the plant kingdom. And so the most famous example of that is the Bt gene from bacteria, which has been very, very important to farmers globally. So this is a genetic engineering technique where you isolate a gene from a bacteria that confirms resistance to an insect. So farmers have been able to massively reduce insecticide spraying. So that type of genetic diversity, I think, will continue to be very important as we go forward. Genome editing is obviously very, very exciting and an important tool, but I think we're going to continue to see more conventional methods of genetic improvement to be used by farmers widely.
Yeah, there's quite a few facets. I mean, many, many different facets there that come together. Now, a lot of people think of science as a scientist alone in the lab, and everyone in science knows that's the far from the truth, that it's a highly collaborative enterprise. And on that note, I want to step back for a moment and just let the two of you be in conversation a bit. So Pam, why don't you kick it off to your longtime colleague?
Okay, and mentor. So Brian, you first taught me about selection pressure, which I think is a really important concept for people that are not in the agricultural community to understand. So I'd like to ask you to talk about selection pressure, kind of explain why farmers can't just get one variety and forget about developing new varieties. Sort of this idea of the ability of pathogens to overcome resistance, how important that is, and also what kind of efforts you and IGI are doing to try to engineer durable resistance.
Great. It's something I've been studying my entire life, I guess, or academic life. And so we talked about taking traits from wild species of plants, and plant breeders have then genetically crossed these into plants. Unfortunately, we usually take one trait at a time. And so you think of a field where every plant is genetically identical out there. And it turns out that pathogens are quite tricky, and that they're able to actually make mutations, and that's due to selection pressure. So anytime in biology, when you put pressure, whether it be antibiotic resistance, whether it be disease resistance, It's a matter of time before the pathogen you'll select for mutations that it can overcome that resistance. I don't want to go into the details of that, but we now understand the mechanisms, how this actually works. But what happens is that a farmer plants a crop, and it's usually only a matter of several years when the pathogen will mutate and overcome that resistance and now cause disease on that previously disease-resistant plant. And the whole process has to start over again, where you have to try to find new genes for disease resistance and then breed those into the agronomic varieties that you need. So it's a constant biological warfare between the plant and the pathogen going back and forth. So to try to counteract this effect, there are many theories in which people are trying to do for durable resistance. One approach that we're trying to do at the IGI is to actually identify multiple disease resistance genes. And so instead of actually inserting or genetically crossing one gene in at a time, we want to bring these genes in as a block. So for instance, if we can bring 6 disease resistance genes in at one time, the chance that a pathogen you'll select for a pathogen that will overcome all 6 of these genes is greatly reduced. Not only that, you can start mixing and matching disease resistance genes such that you can distribute them within the field and not have all the plants genetically uniform. So this is the power of gene editing or CRISPR technology, is that we can introduce genes that we can't do by classical breeding into plants. And so making these novel combinations of disease resistance genes, we hope to be able to actually make more durable resistance.
Great, thanks.
Brian, as you've watched, I wanted to let you toss a question back to Pam as you've watched
Sure,
your own proteges rise and flourish in her career.
No, it's always great to have students like Pam. I think a question I'd like to have you ask to you is that, we talked about GMOs, we talked about gene editing, and I think one of your more recent publications is a great experiment where you actually made rice plants that actually would help cure blindness. So if you could compare and contrast your efforts with GMOs with those of gene editing for traits that you're interested in developing. And maybe draw upon your book, which I think is something that people should read. I'll give you a plug for your book also.
Okay, thanks. So for the carotenoid rice, so maybe just to go back a little bit, the audience may be familiar with golden rice, which is a really tremendous, exciting breakthrough by Peter Bayer and Ingo Petrikis a number of years ago now. And so the Rockefeller Foundation supported them to develop rice that had higher amounts of ultimately vitamin A. And the reason is that vitamin A deficiency is a very serious problem in many parts of the world. It's estimated that 500,000 children go blind every year and half of the children die. So this is obviously a very serious situation. It's especially prevalent in areas where families don't have a diverse diet. They're eating rice 3 times a day. They don't have access to other types of foods that are nutrient rich. And so the idea was to engineer carotenoids, which are the precursor of vitamin A, into rice. And they've been very successful. And there is a rice variety that's been approved for food consumption in many countries now, including Bangladesh, as well as developed countries, New Zealand, United States, Canada. And I think it's the last regulatory hurdle, and hopefully it'll be in the hands of farmers soon. But we got very interested in this and thought it would be a nice pilot test to try to use genome editing to introduce this particular cassette into rice. And we chose it obviously because it's very important for socioeconomic reasons, but also it gave us a nice example. So it's big, it's like 5.2 kb. So Oliver Dong in my lab, really, who carried out the most of the experiments, was able to identify what we call landing pads. So a region of the rice genome that when you insert a new gene will not disrupt other essential gene function. And so he was able to identify a number of landing pads and was able to insert this carotenoid cassette into 2 regions of the genome. And he was able to come up with these golden rice. So they're golden. They have high levels of carotenoids. So it's sort of a proof of concept that we will be able to use genome editing to introduce agronomically important traits to specific regions of the genome.
Pam, just quickly, some of our listeners remember a cassette as something you put into a Sony Walkman, and some have no idea what we're talking about. What does a cassette mean in genetics?
Okay, so a cassette. So back in the day, we'd put a single gene into a plant, and we'd study how that plant grows and how it resists disease. But now scientists are able to, in a sense, reconstruct biosynthetic pathways. So the endosperm, the rice grain that we eat, does not produce any vitamin A. And colleagues over the years were able to identify really just 2 genes that needed to be added. So in this sense, a cassette is a cassette of 2 to 3 genes that you can insert as a unit into the genome.
Thank you. So since we're talking about rice, another follow-up, and in fact, we'll turn now to some questions coming from the audience. So thank you all very much. And if you are listening, YouTube Live and Facebook Live, you have a chat window there. You can put in questions, and I'll just say up front, we don't get to all of them, but we will do as many as we can. So there's a very broad question here, Pam, and it's also kind of 2-part. One is, as you pick problems to work on, and I'm paraphrasing, as you pick problems to work on, what role do the social equity questions play for you about, who would benefit from this work? What need is it addressing in a broader societal context? And then the specific follow-up from that is, once you've done your work, like with the rice example, how does it get from that research lab out to a subsistence farmer? And what's that pathway? And is it affordable? And once you get to the other end, is that an affordable product that really can be used widely around the world?
Yeah, thanks. I think social equity really drove my interest from the start. And my father's a immigrant, mother grew up in the Depression, so we really grew up with this idea of trying to give back. And I was attracted to Brian's lab because he worked on disease resistance, and very, very excited about the idea that farmers can plant a seed, a genetically improved seed, and then may not need to spray any insecticides or fungicides. So that whole environmental aspect, social equity aspect, really, I would say, has driven my work. And working on tomato and pepper in Brian's lab was really fantastic, but I decided that I wanted to work on a food staple. So that's why I switched over to start working on rice, which was also becoming a model organism at that time, about 30 years ago now. I think I got my PhD right when PMB was coming together. And so then picking what diseases and traits to work on, it was obvious to start working on disease resistance because of the training I had in Brian's lab. But then a colleague introduced to me the very serious effects of environmental stress. So that's why I started working on the submergence tolerance. And I would say that it is really critical to have sort of a pipeline. So if you're able to do something useful, and we all try to do our best, and you don't really know what's going to work and what's not going to work, but to build that pipeline early. So for the submergence tolerance work, it was really brought to the attention of scientists from farmers. I mean, they were losing their crops in flooding. And the International Rice Research Institute is part of a very important international nonprofit program that is focused on putting improved seed in the hands of farmers. So the International Rice Research Institute already had a whole set of collaborators in Bangladesh and India that were able to trial very quickly, with Dave McKill's leadership, trial the new varieties that were being developed. And then the seeds then are just put in the hands of farmers through the normal network. So in in many less developed countries, including Bangladesh and India, rice farmers will go to their national germplasm centers, and they will get the seed from those centers. So in the case of submergence tolerance rice, there was no need to develop any kind of new distribution system. And because the seeds are not regulated, marker-assisted breeding is not a regulated technology, it was really quite, that part was quite easy. Well, I shouldn't say easy, because they still had to bulk up a lot of seed. The Bill and Melinda Gates Foundation did provide funding to the Bangladeshi Rice Research Institute, the Cuttack Rice Research Institute in India, and the International Rice Research Institute to help breeders bulk the seed and distribute to farmers in those areas. And then I should say, Kyle Emmerich and other colleagues in your college did a really important study showing that the benefits of planting sub-1 rice accrued to the most disadvantaged farmers in the world. And that was a really fascinating study. And it's because in India, there's a caste system. And historically, for hundreds of years, the lower caste farmers have had the most flood-prone land. And so they're really seeing the major benefits.
Oh, that's a fascinating connection. And also, many of our listeners will know that California does not produce vast quantities of wheat and corn, but we do have flooded farmland and a significant rice industry. So right there in Davis, you're in the heart of a fairly important rice growing region as well. So that an important connection to across to other parts of the world where it becomes the main staple. Brian, I'm going to switch gears a little bit, again, picking up from our listeners. I think it's fair to say you've been in this business for a long time. You've had students, many generations of students. And again, to paraphrase from the question that came in, the question is, what advice do you give students entering the field now about both the technical skills they need, as well as like what we'd call the soft skills? And how has that advice changed? How has your mentoring had to change as the science has changed over the decades?
It's a good question. I think the way I have always approached science is really to ask biological questions and then basically try to find technologies that will help me to basically solve those problems. I think the best advice I can do today for students is that the interface of computational biology and molecular biology are more paramount than ever. Being able to, because what's happening is that there's a massive revolution in gene sequencing technologies, and we've created these massive databases. And students have to be able to manipulate these databases and to be able to write code and scripts to be able to deal with these large databases. So I really encourage students to get trained in computational biology and being able to bring those two systems together. Also, I think it's important for students. What I find today most interesting, most students really want to make a difference in the world. So they want to do basic research, but they want to translate that basic research into solving problems that have an impact on humanity. And I think instilling those particular traits into students has been important as we've gone along. Again, I think having a passion, show people how you have a passion for science and how important that is in accomplishing your task. And those are probably the most important things.
So Brian, you may not be able to see this. We knew that we had a slightly unstable web connection to Pam, and I think she may have dropped off for a moment. So we'll hope she drops right back in. So let's just continue, the two of us. Going back to the traits, you talked about how climate change will impact disease, which is the area you've spent your career in. Can you say a little bit more about traits, maybe non-disease traits, just since you're in these discussions widely? What are key traits that gene editing might be able to help with, specifically in relation to our now very focused attention on climate change as a challenge for agriculture?
Right. I give an example from work in my laboratory where Nicholas Karavolius is a graduate student of mine, and with support from the Open Philanthropy Project. We're studying stomatal density in rice with the idea that we can regulate a particular master regulator called stomagen, where we can have an expression from almost very few stomates to overexpression of stomates. And it's well been established in the literature that if you find this sweet spot of stomatal density, that you can actually make plants more drought resistant. So you can use, not only is CRISPR important for inserting things, it's a research tool. You can actually make allelic diversity of genes to be able to study traits. You can also, as many people may or may not know, that many genetic traits in plants aren't single genes. They're what are called quantitative trait loci. We don't need to go into the details of that, but needless to say, there are many genes that contribute to a trait. And using CRISPR technology, you can dissect out which of these genes that are really important. So there are many of these QTLs for drought tolerance. And so we're trying to actually think about ways in which we can use CRISPR to identify which are the important genes in these quantitative trait loci. So lots of things to do and a lot of challenges out there, but we think that making drought resistance is going to be really important.
So we've focused so far mostly on the drought issue and the flooding, drought, and disease. But in terms of the discussion about sustainability, one of the key challenges is the inputs into agriculture, and two of those inputs are, of course, fertilizer and pesticides. And I believe Pam is back. Pam, we noted your brief Wi-Fi dropout, but you're back. So tell us a little bit about the current status of research and the role of gene editing and genetic technologies around specifically addressing this issue of the inputs to agriculture and the problems that arise from both high nutrient inputs as well as pesticide use.
Yeah, as I mentioned earlier, the BT trait has been really phenomenally important for farmers in reducing their insecticide use. So just for an example, eggplant, which is the most important vegetable in India and Bangladesh, is also very susceptible to insect pests. And farmers in Bangladesh were spraying their crops 3 times a week, sometimes even every day, to try to control this particular pest. And especially in less developed countries, sometimes the insecticides are not really controlled or regulated. And so it's very, very harmful often to farmers and their families, especially when they don't have proper protection. So scientists at the Bangladeshi Rice Research Institute in Cornell were able to engineer eggplant to carry this BT gene, and were able to dramatically reduce insecticide use, often down to 0. And so I think we need to keep in mind the importance of reducing insecticide use. And sometimes I hear sort of a little bit of confusion in the public that don't fully understand that the idea of the BT trait is to reduce the use of insecticides, some of them which can be really quite toxic. So that's why traits like BT will continue to be important. There's a lot of scientists that are working on nitrogen use efficiency and phosphate use efficiency. I think these will be very important. I don't know, maybe Brian knows, but I don't think any of these traits are out really in the field in the hands of farmers. But the general idea is that if plants are able to use these really important fertilizers more efficiently, there will be less runoff into streams, into our ecosystems, and into the Gulf of Mexico, and many other areas which are, there's a lot of nutrient runoff and pollution. So I think genetic technologies will be important. Of course, management is important as well, to try to add those inputs sparingly. So it's really a combination of farmer practices and genetically improved crops that are going to be critical for addressing these important problems.
So there's a question here, which it follows in part on that, and that is that one of the really novelties that arose, frankly, in the last 30 years is the idea that living organisms and genes can be patented and that they have intellectual property around them, and which is not a lot of us wouldn't be intuitive if we think of a lot of this as sort of products of nature, but these are very much an intersection of a natural product and the ingenuity of crop breeding. And so my question, and I'll start with Brian, but maybe I'd be interested in both of you, is especially when you're thinking about these things like these equity issues, where does the intellectual property side come in terms of what does it mean to own these discoveries? Of course, there's a lot at stake and potentially a lot of financial consequences in stake in terms of both the inventions and also those who are the downstream users. And is gene editing changing at all the landscape in terms of intellectual property and agriculture?
You know, that's a great question, and it's a very complex issue, as you could well imagine. Obviously, there are traits that you can gene edit and you can patent these. It really depends on what the patent holders of this, what they want to do with licensing these particular technologies. I've pushed at the IGI that we would not take any of our discoveries and would not exclusively license them to everybody, so they would be widely available. And furthermore, if they're for developing countries, they would be given freely to those countries. And many companies actually have made that pledge that they would actually give their intellectual property to developing countries. That's important. So maybe the developed country, which they might be able to afford to acquire these licenses, but in the developing world where they can't, I think would be very important. So it's a complicated matter. There's lots of intellectual property out there, and even the whole CRISPR patent situation is still being debated and resolved. It hasn't been resolved totally yet.
Pam, anything to add from your perspective?
Yeah, I think, you know, there are specific examples. So for example, Golden Rice, which was developed, it was funded by a nonprofit Rockefeller Consortium, but they borrowed different, you know, tools and parts from Syngenta. And what ended up happening is Syngenta released all their rights. And so Golden Rice, that is now being, hopefully will soon be grown in Bangladesh, Philippines, and other parts of the world, will be completely in the public domain, which is really very important if you're talking about very poor farmers. So they'll be able to plant the plants, harvest the seed, and replant. So there's no intellectual property restrictions. And I think that is critically important in the less developed parts of the world. The same with this emergence tolerance gene. We made the decision to be sure that that gene was publicly available. So there's no, we published it, so there's no patent, there's no UC Davis patent on that gene. So that means if anyone wanted to use it in genetic engineering, they could use it. But also, if you're just using marker-assisted breeding and not using any of our, you know, genetic sequence, that also would be in the public domain. So I think it's very clear in less developed countries that there has been and there will continue to be a lot of push to be sure those tools are in the public domain. And it's a different story in the United States and Europe, where almost all farming is for profit, whether you're an organic farmer or a conventional farmer. So the landscape's a little bit difficult there. I think one thing maybe we can talk about if we have time is this idea that it's the gene itself is really where the patent is held, but it's actually often the germplasm. So corn breeding has been developed for 70 years. So there's very valuable what we call germplasm, which is just the genetic background that's used to make hybrids. And those are very, very valuable, even independent of any new genes that are being added. And so there's a lot of interest in and concern about the genetic background itself being patented. And I don't know if Brian or David have anything to add to that, but it's certainly an ongoing debate.
No, Brian, anything to follow up on that?
No, I just, like I said, it is an ongoing debate, and you know, hopefully that good rationale will prevail in trying to make these technologies move forward. And so, yeah, I don't have anything more to say about it. Sorry.
So, so one thing I really hear you saying is that it matters where the funding comes from. Foundation funding has really been important here, and now, PMB has had a long history of very close collaboration with some private sector players in biotechnology. So as you've watched this unfold over several decades, I'll ask Brian, have you is that relationship really changing? Do you see a shift in the nature of the relationship between university researchers and private researchers, and then this interface to making things available around the world?
It's interesting. I gave you the example of cacao, where Mars Corporation is not interested in holding any intellectual property. Their major motivation is to actually have a supply of chocolate. So they're willing to give funding to develop technology so farmers can produce the chocolate that they can use. So I'm not sure all the other companies have the same rationale behind that, but it's definitely, but a lot of the companies I know that if they have intellectual property like Corteva, which used to be Pioneer Hybrid, they have actually allowed their technologies to be used in the developing country. They will give the rights to those in developing countries. So I think on a case-by-case basis, hopefully we'll be able to accomplish this. I think it's important that we democratize these technologies.
That actually, I mean, that's an incredibly important sentiment there, and also just the idea that there are no single silver bullet statements or answers across all these things. That it is very much case by case, and we need to look at each case on its own merits, I would say. In the last few moments, I'm going to turn it a little bit more, maybe a little lighter and a little bit. I'm going to say a personal question. It's not very personal. I have a great question from the audience for each of you. Who are your scientific heroes?
Well, Brian, of course. There's the obvious answer. It's the only answer in this setting.
Well, that was a setup. But Brian, how about you? Who is the?
I would say that my professor, my PhD professor, was a real intellectual. He's still around. Nicholas Panopoulos was his name, and he really taught me how to think in testable hypotheses. And this really has stuck with me my entire career. And he was a great mentor, and I can't say enough what an influence he had on me, how to approach science and to ask important questions. And one thing he always says, if you can think of an experiment, and if it's not inexpensive, just do it. Don't talk yourself out of it, because you'll often find that it may not work the way you want, but serendipity comes into play, and you will find something totally new. So I would say Nick Panopoulos for me.
Well, and Brian, that speaks to something that, again, people who are not inside the scientific research community may not fully realize how powerful that mentor-student relationship is, and it shapes our entire careers. And I think all of us could tell that story of the influence of our advisors and what a difference it made. I want to give you each an opportunity for some closing thoughts, and specifically, this is a 30th anniversary event, and those who know our history at Berkeley, PMB came together in a merger and reorganization. It wasn't the invention of biology, it was just the founding of the PMB department in its current form. But what about the future? If you could look ahead to the next 30 years of plant biology, plant breeding here at Berkeley, at Davis, what's on the horizon? And of course, this is one of the questions for students entering the field, thinking what their career might look like. Brian, Brian, go ahead and start.
Okay, I can just start it. So I think that obviously I've been at Berkeley before the PMB department started. And I think what's really been important for me as a faculty member in PMB is the great colleagues that we've had that really form. Berkeley is, again, a great place that has attracted amazingly good graduate students and postdocs, and I've benefited from being able to attract those students and postdocs. But also, I would generally say across campus that I've developed collaborations with people outside the department. So I think as you move forward, you just can't work with just a few people in your own department. You have to reach out because there are new technologies. I'll give you an example. We've just recently used cryo-EM technology to really get an important complex of disease resistance immune receptor out. And that's with the collaboration with the Ava Nogales' lab and Raul Martin, who's the graduate student. So being able to go out and to really interact with other members, not only on this campus, but UC Davis, we have a lot of close relationships with the great plant sciences at Davis, at Stanford, UCSF. So I think as you go forward, Berkeley is in a great place because you have these four major institutions. You've got, UCSF, Stanford, Berkeley, Davis, and it's all within a 70-mile radius or 100-mile radius of each other. So it's really, Berkeley's a great place to come to do science.
Thank you. And Pam, some closing thoughts.
Yeah, well, I really appreciate being invited back. Being a graduate student at Berkeley was really a fantastic experience. You know, I gained from Brian's mentorship and also Nick's mentorship, who was there when I was there, and also so many fantastic faculty in the program. I know many of them are retired. Lou Feldman, Dick Malkin, Steve Lindau, Patty Zambrisky, John Taylor. I mean, Bob Buchanan, we had a really fantastic group of professors that were mentoring us. And so it's been a really excellent to be back. And, you know, I concur really with what Brian said. You know, as you go forward in your career, you continue to meet fabulous scientists and many scientists that are willing to help you in an area that you're completely unfamiliar with. And that's one of the great things about being a scientist, because you're always learning. And so thanks very much for giving me the opportunity to join you today.
Well, thank you, Pam. Thank you, Brian. Thank you to everyone who's listening for joining us today. We hope you'll come back for future events as part of PMB's 30th anniversary. In the meantime, stay safe and best wishes.